Microbiology students, welcome back. We are looking at Lecture 9, which deals with the topic of microbial genetics. So a really important part of this lecture is the core content review. And so I've made a video or two there along with some other videos, but to understand this, it's really critical that you review the structure of DNA and the structure of RNA and the processes that are used within cells to allow the synthesis of RNA from a DNA molecule, and then also the synthesis of protein from an RNA molecule. So those processes are called transcription and translation, but then also we review DNA replication as well. So it's understanding the structure of nucleic acids is one topic, and then really three fundamental processes that nucleic acids are involved with, which is copying DNA, that's replication, making RNA, that's transcription, and then also making proteins, which is translation. So I'm going to review a little bit of that now, but again, it's not going to do it completely justice. And so it's a topic that you've had in intro bio and in other classes, and so I have those resources as a review. But real quick, the structure of DNA is really critical to understanding much of what we're going to be talking about. And so here is an image of the structure of DNA. And so there's different aspects that are really critical to the understanding of this. So first is we call nucleic acids, which includes DNA, they are polymers, meaning they have building blocks that are linked together to form their structure. And so the building block of all nucleic acids are nucleotides. So this is the building block. And what a nucleotide is it's composed of three different parts. And so there's a sugar, and the sugar in the nucleotides to build DNA is called deoxyribose. There's at least one phosphate, but it can be upwards of 3 phosphate groups. And then there's also what's called a nitrogenous base. And the nitrogenous base is going to be really critical because it's this base that differs in the 4 different nucleotides. So there are 4 different nucleotides that can be used as building blocks to build DNA because there's 4 different and unique bases. And you probably remember them as having just single letter abbreviations. So there's an A, there's a G, there's a C, and there's a T. So depending upon which base is linked to the sugar, is then going to determine which of the 4 nucleotides is present. So here's an example of a nucleotide. Here's the sugar, deoxyribose, and then here is one of the nitrogenous bases. It happens to be the G, or guanine, and there's the phosphate. So that piece is the phosphate group there, surrounded by the oxygens. So there's the base, there's the sugar, and there's the phosphate. So that would be one nucleotide, but it's linked together with a neighboring nucleotide. So again, here's the sugar, here's the base, and here's the phosphate group. So this would be a second nucleotide, and they have been linked together with a specific bond. It's called the phosphodiester bond. And so that's, I'll just draw that right there. So that is the phosphodiester bond that we're talking about. And so that's what's linking all the nucleotides on a single strand. And so this purple line represents this purple line right here. And so this would be one strand of DNA. And now it's just kind of abbreviated. This has, because the nitrogenous bases are what provides a unique information, that's all we really show here. So this nucleotide has a T attached, this one has a G, this one has an A, that's a G. And so frequently when we look at nucleic acid sequences, we just write these single letters in a row to indicate what would be along this chain or strand of DNA. Now, DNA typically doesn't exist as just a single strand, but it exists as what we call a double helix. And so double just means that there are 2 strands interacting, and those 2 strands kind of twist together like a spiraling staircase, as shown here in this picture. So not only do they form this helical structure, but we also know the 2 strands interact in a very specific way. And we call this complementary. And there's kind of 2 parts to this complementary nature of how strands interact. The first is when you look at the 2 strands, they run in opposite directions. So what we mean by that is every strand of nucleic acid has a certain directionality to it. And so if you look at one end, it can be designated the 5' end. And if you were to follow this same strand all the way to the other end, the opposite end has what's called the 3' end. So that just has to do with how we label things in organic chemistry. And so this is the 3' end. There's nothing attached at this 3' end, and there's nothing attached to this 5' end. So when you look at this second strand, we notice that it's running in the opposite direction. So this has the 5' end here, but the other strand, note it has a 3' end. So we call that anti-parallel. So that describes in part how the two strands interact, but the other really important piece is that the bases on each of the two strands actually orient towards each other. And so one way that people kind of draw this is like a ladder. So they're kind of rungs of a ladder across. And so the bases are actually oriented towards each other. So those would be like the rungs of a ladder. And whatever base is on one strand, there's a certain rule as to what the base is going to be on that second complementary strand. And this is what we call base pairing relationships or base pairs. And so if there's an A on one strand, there's going to be a T on the other. If there's a G on one strand, there's going to be a C on the other. And so when you look at this, you always have them pairing up with that rule in mind. So there's a T, there's an A, here's a G, here's a C, here's an A, there's a T. So you always have T's and A's and G's and C's. And so you can follow this all the way down. They've color-coded it, and so you can see that pattern holds true all the way through the length of this. And so while the bonds that are along a single strand that are holding the nucleotides together, those are called phosphodiester bonds, these dotted lines here are what explain why these two nitrogenous bases want to pair with one another. And those are called hydrogen bonds. I'm just going to write H bond here. It's on the bottom of the slide. So it's right here, hydrogen bonds. So that's the kind of quick review of what DNA's structure is. There's one other concept that I want to review before we move on to kind of new material. And that is what's called the central dogma of genetics. And so this is how information is used across all cells. So whether this is a human cell or a bacterial cell, there's a certain flow of how information is passed between molecules. And so we were just talking about the structure of DNA, and we called it a double helix. So this is simplified a little bit. They don't show it twisting, but they do show the anti-parallel nature of it. These would represent the rungs of the ladder, and so those would be the base pairs. And again, if there's an A on one, there would be a T on the other. So this is simplified, but it's double-stranded DNA. And that molecule within any organism, so the DNA of a given organism, is going to have all the information that's needed for the survival and the growth and all the aspects that's needed for that organism to live. And so to kind of boil DNA down into a very simplified explanation is, I frequently use this term, it's the instruction manual. And so if we take human DNA, in that molecule, in the DNA, the human genome, there is all the information needed for human survival. So regardless of what cell type you're talking about, the DNA is going to tell heart cells how to be heart cells and what they need to function to do, or skin cells, or neurons, or liver cells, and so the list goes on. And so within that DNA, there's all sorts of information. So what this central dogma shows is how that information is converted to other molecules that can carry out the important functions that exist within cells. And so one process is simply copying DNA. And so DNA replication is the process by which, how can a cell take one instruction manual so the complete set of instructions, and then make a duplicate copy of it? Well, the answer to that is DNA replication. That's how it's achieved. Then there's also the process of not copying all of the DNA, but instead taking a discrete region of it and making not a DNA, but rather an RNA copy. And this is called transcription. And so making an RNA copy of a specific DNA sequence is called transcription. And the reason why that can be beneficial is that RNA can then go and interact with the protein synthesis machinery in other parts of the cell and actually make these really important functional molecules called proteins. And so the information that's stored within this RNA can be used in the process of translation to make proteins. And proteins are really the workhorses. They are the enzymes, they are the structural components that help hold cells together. They're hormones, so they're signaling molecules, they're proteins on the surface to receive information from other areas. So proteins are really critical. And so this central dogma helps show how information that's embedded or stored within a DNA molecule can be converted through this process of transcription and translation to proteins that then have some function. So kind of recapping this, DNA controls all the genes, DNA contains all the genes for controlling an organism, so it's that instruction manual. It is possible for some of these genes to be always on, meaning this process of transcription and translation is always going on. So if you think about some of the proteins we might need all the time, like those involved in glycolysis and Krebs and electron transport, those are so critical to our metabolism that they are basically, those genes are on all the time. Now, there are other genes that are said to be regulated, or they're expressed only at certain times. And so this brings us to the next several slides that explains what we call gene expression or gene regulation. So think about the lights in your house. You don't have to have your lights on all the time. You turn them on when you need them. You turn them off when you don't need them. And so this process of transcription and translation can be turned on and turned off depending upon what the need is of the cell. And so this process of gene expression is going to explain if RNA is made, so is this transcription occurring? And then if that transcription is occurring, then we can get the protein. But as you might guess, if this is turned off, no transcription means no RNA. If there's no RNA, then you won't get proteins for that particular gene. So gene expression is the topic we want to move on to, and there's two examples that we're going to look at. And so these are two questions before we move on. So if there's a bacterium, the question reads, should a bacterium make proteins, this should be an S here, for lactose metabolism if there's no lactose? So if you are unfamiliar, lactose is a specific type of sugar. It's called a milk sugar. So it's a disaccharide, so two monosaccharides linked together. And so bacteria have the ability to actually make enzymes that allow them to break down and use lactose as an energy source. So this question is posing, does it make sense for a bacterium to expend energy making the enzymes and other important proteins to break down lactose if there's no lactose for it to use? It would kind of be a wasteful situation. So as you might guess, bacteria don't make lactose metabolism proteins if there is no lactose present. Here's another example, and again, it's based on a question. It says, should a bacterium make more arginine? And arginine is one of the 20 amino acids. So arginine is an amino acid. So I'm just abbreviating that, amino acid. And if you don't remember, amino acids are used to make proteins. So I'm just going to abbreviate that too. So amino acids are the building blocks of proteins. So as this reads, should a bacterium make more arginine if there's already sufficient arginine present? So this is similar to if you have already purchased, say, a case of macaroni and cheese from Costco. So you have whatever that holds, 24 boxes of macaroni and cheese. It doesn't make a lot of sense to go and buy more macaroni and cheese because you have this stockpile. And so the bacteria are in a similar situation. If they have enough arginine already present, does it make sense to spend resources and energy to make something they already have sufficient quantities of? And so because of this, there is a regulatory mechanism that helps the bacteria achieve each of these desired outcomes, so that lactose metabolism genes are turned off when there's no lactose, but then these arginine metabolism pathways are turned off if arginine is present. And so that's what we're going to look at in bacteria. How do they actually achieve these two desired outcomes? So part of the answer is unique to bacteria, and these are called operons. So this is not exactly how gene expression works in eukaryotes, but it does in bacteria. And since this is a microbiology class, we are very interested in understanding the genetics of bacteria, because it's going to help explain how some of them can be virulent, or how they can cause disease. So we'll see that in the future. And part of how their genes are expressed or regulated is in this functional unit that's called an operon. And so what an operon is, it's a coordinated set of genes and that function as one regulated unit. So basically, when an operon is turned on, you get several genes turned on at the same time, and you have this this function that works across the operon. So, for example, there is something called a lactose operon. That's going to be our first example, and that has 3 different genes in the operon, and all of those genes are going to be important for lactose breakdown or lactose catabolism. And so if the organism is going to be able to functionally use lactose, then all 3 of these genes need to be on at the same time. And so to achieve that, they're regulated as one operon. So all of them are on at the same time, or all of them are off at the same time. So that's what an operon is. The other example is the arginine operon. So we're going to look at that as well. And so again, it's a series of genes. They're going to be turned off and turned on. So if we go back to our light analogy, if you have, say, recessed lights in your ceiling, you don't have a switch for each individual light, but you have them tied together so that when you hit one switch, all of the lights that you want tied to that one circuit are going to be turned on. And so that's kind of what is happening here with these genes. You have them functionally tied to one operon, so all the genes are on at the same time, or all of them are off at the same time, and you don't have to individually turn on or off these genes. You just do it as an operon. So there are 2 classifications of operons. So one is called inducible operons. So that's the one we're going to talk about first. And the other are called repressible operons. So the difference between them is kind of what their base level state is. So inducible operons are by default turned off. And so what that means is they need some trigger to be turned on when it's appropriate. So these are inducible operons are typically involved in catabolism. So if you don't remember, catabolism are going to be those breakdown reactions. And so that means you start with big molecules, and they are reduced through catabolism to smaller molecules. And usually, sorry about that, smaller molecules, usually you get energy at the same time. So one example of that is lactose catabolism. So again, this is a sugar that can be broken down to generate energy. And so the lactose operon is inducible because it's turned off in bacteria, but it can be turned on when the need is generated. Okay, so that's an inducible. And so we'll see this lactose operon a little bit to show how it works. A repressible operon is just the opposite. It's usually on, so by the default state is it's on, and it's turned off excuse me, when it's not needed. So it's on, but then something will trigger it to be turned off when it's no longer needed. And these are going to be more for anabolic reactions. So anabolic reactions are going to be where you start with something small, and by using energy, you can link those smaller things together to form bigger molecules. So that's what anabolic reactions are. And so the arginine operator is the example that we're going to use for this. And so we'll do that. We'll do these two in order: lactose operon first and the arginine operator second. So the lac operon, short for lactose operon, we already said it's inducible. And again, what that means is it's normally turned off. So that's the default state, is off, and it's going to be turned on when it's appropriate. The way that that's going to happen is there needs to be the inducer molecule. When the inducer molecule is present, that's going to signal the operon to be turned on. So it's normally off, but when the inducer is present, it's going to trigger the turning on mechanism. And there's several features of the lac operon. There is the regulator. And so this is a gene that's going to make a protein that's called the repressor. And this is really the off switch. So the repressor, we're going to see momentarily, but the repressor, you can think of this as the off switch. And then there's what's called the control locus. That's a fancy word for control region. And this houses two specific bits of DNA. It's called the promoter and the operator. The promoter is where a certain enzyme binds and starts the process of transcription. So RNA polymerase is an enzyme, and it's responsible for making RNA. So it's kind of in the name. Polymerase, we see it's an enzyme based on that ending, and so it's going to make an RNA polymer. That's what the name is. The operator is where that other protein that I just talked about where repressor binds. So I said the repressor is the off switch, but for it to serve as an off switch, it has to physically bind to this operator and basically block the action of RNA polymerase. And so the picture will show us in a little bit, but the control locus has these two DNA regions, the promoter and the operator, which we'll see momentarily. And then the structural locus is the genes of the operon that were going to be made when the operon's turned on. So in the lac operon, there are 3 genes, and all 3 of them are used in the process of lactose breakdown or lactose catabolism. So those are the definitions. So let's take a look at this. So these are the different parts that we mentioned for the lac operon. There's the regulator. This is the control locus, as we saw. And then after this, that's the structural. I'm just going to abbreviate because this is a little hard to write. That's the structural region. So those are the 3 parts that we mentioned before. And so what the regulator does is it is a piece of DNA that allows for the production of this repressor protein. So remember how DNA is used to make proteins. It's through the process of transcription and translation. And so this repressor is always being made. And in a normal environment, it can bind to that operator and sit right here, which is going to serve a purpose, okay, which I'll explain in just a second. And so when this repressor is bound to the operator, the gene is off. Okay, so that's the off switch. It's those 2 things working together. Repressor bound to the operator, the gene's turned off. And the reason why that occurs is the polymerase will bind to this promoter, but it can't really move in this direction to read the DNA to actually transcribe it. So it's basically blocked. And so there is no transcription, as this shows, there's an X here. There is no transcription of these structural genes in the operon. So even though the DNA is present, there's no RNA being made, and if there's no RNA, as I said, there's no translation. Okay, so that's what we mean by this operon being off. And so this is the normal state. And so this is called an inducible operon. So the normal state, as we mentioned, is in the off position. So now what we want to look at is what happens to turn this on. And so there needs to be some signal to turn it on. And in this case, if they're called inducible operons, it's the presence of what's called the inducer that's going to trigger the turning on of this gene. And so in this case, I'll back up and read this. So this is kind of the state that we described first. The regulator is always on, so this gene's always on, and that's going to make the repressor. The repressor binds to this operator sequence and blocks the transcription of these structural genes. Okay. If the inducer, in this case, it's lactose, is present, then that lactose can physically bind to this repressor protein. And upon doing so, the repressor is actually going to change, it's called change its conformation or change its shape. And that change means the repressor no longer binds to the operator. And so this is going to leave. And so there's no longer a repressor attached. So we said it was that situation that made this gene to be turned off. So once that repressor is no longer interacting, we would say, oh, this operon is now turned on. And what that means is, with the repressor gone, that RNA polymerase, it binds to the promoter, and now there's nothing blocking it from moving in this direction and physically interacting with all of these structural genes of the operon. And so what that leads to is transcription. So you get mRNA. So there's the I was just looking at this, and the wording here is not the best now that I'm looking at it. You have the transcription, which makes the RNA, and then you have translation, which gives you the actual proteins. And so when this gene is turned on, you ultimately get all three of these important proteins that are needed for lactose catabolism. So they function as one unit, and that's what an operon is. The last thing that's shown here, as lactose is depleted, and it will be depleted because it's being used as an energy source. So as the cell starts to break down lactose, the concentration of lactose is going to diminish. Then what's going to happen is the repressor is no longer going to be able to interact with lactose as the lactose starts to disappear. And so once the lactose starts to diminish, then the repressor protein can bind to the operator again, and therefore it's going to shut off the operon again. So we go back to that off state. So no inducer, I should word it, I'll say, so if there's no inducer, change that to an R, so no lactose present, no inducer present, the gene's going to remain off, or more specifically, the operon is going to remain off. So that's how the lactose operon operates, or the lac operon for short. And so there's some similarities to this other operon, but it is what's classified as a repressible operon. And so that means the default state is on, and so it needs some trigger to be turned off. So there's a lot of similarities to the structure. There's going to be a regulator, which makes a repressor, so that's the same. There's a control locus that contains the promoter, where RNA polymerase binds, and an operator, which is the repressor I'm try to sneak this in here. The repressor binds that operator. And then following this control locus, there's the structural locus as well. So let's look at this one. So similar setup. Here's the promoter. Here's the operator. So this is, again, the control locus. And then here are the structural genes. So the normal state, the default state, is the repressor, even though it's present, it cannot physically interact with this operator in this default state, and so it's not bound. And with the repressor not bound to the operator, then RNA polymerase is free to interact with this and actually make these structural genes through the process of transcription and then translation. So in this case, the regulator is always being made, but the repressor does not bind to the operator, and so because of that, it does not block the expression of those structural genes. So another way of saying this is this operon is turned on all the time until when there is the presence of what's called a co-repressor. So we don't call this an inducer anymore, we call this a co-repressor. And in this case, the co-repressor is arginine. And so as arginine accumulates, and so again, arginine is an amino acid, and these enzymes are responsible for making arginine, and so if they continue to work, you're going to get more arginine being made, because that's what these enzymes and products of this structural locus are. The enzymes synthesize arginine, so you're going to get arginine accumulating. So if arginine accumulates above a certain point, it can serve as what's called the co-repressor, where it's going to bind to what was the inactive repressor. So here it's inactive, but once arginine binds to it, it's going to, again, cause a conformational change or a shape change. So if you look, this shape looks a little bit different than this shape, and that's because the co-repressor arginine is now bound to it. And in this state, the repressor becomes active, and it can bind the operator. So there it is, binding the operator. And that, as we said before, turns off the operon, meaning RNA polymerase cannot get to these structural genes, so there's no transcription of them. And if there's no transcription, then there's no translation, so the proteins aren't made. And so effectively, the operon is turned off. Okay. So in this case, the gene's on until the co-repressor reaches a certain concentration. And once that occurs, it binds to the repressor, causing a shape change where the repressor can now interact with the operator. We can also look at the reverse of this. So the operator will go back to its original state as the arginine concentrations start to diminish again. So in this case, the bacterial cells can use arginine to make other proteins because it's an amino acid, so it's a building block of many important proteins. And so over time, these arginine molecules can start to be used to make proteins, and so their concentration will diminish. And as it lowers in concentration, then you will no longer have arginine bound to this repressor. And so then when arginine is no longer present, the repressor is going to go from this state where it's active to the state where it's inactive when there's no arginine associated with it. And so without the co-repressor, this repressor can no longer bind to that operator, and then you'll have the gene turning back on. So those are examples of how operons operate within bacteria. We have the lactose operon, which is an inducible operon, and now this shows us the arginine operon, which is an example of a repressible operon.